Hello guys, I am Shubham from ShubhVed Tutorials. This video is about translation synthesis. So before we start, subscribe to our channel and press the bell icon to get notified whenever the video is out. So let's get started with the video. We are discussing the different repair pathways and this video is about translation. What is translation? As we saw in the non-homologous end joining, this is also a mutagenic process. So a mutagenic repair pathway. What is mutagenic? Basically, it leads to mutations. Okay, so mutations are produced, right? This is a mutagenic process. And what is it basically? What we can understand from the word translation means what? It bypass the translation. Bypass the translation, sorry. Bypass the lesion. Or we can say damage. Okay, how it bypasses, we will see further. So the lesion or damage is bypassed. The polymerase moves over the DNA. If there is lesion there also. Okay, means what? Suppose we say like that. I will draw your DNA strand. See, this is a lesion, right? So what will polymerase do? Polymerase will move towards it. Polymerase will move towards, right? It will not repair. It will not repair the lesion. It will just move forward. Okay. So this is basically a synthesis process. We are seeing translation synthesis. Means what? Synthesis is taking place. In normal repair pathways, what happened? The replication fork stops. Okay. If there is a lesion or damage, that has to be repaired first. So replication is stopped. In this case, replication does not stop. Replication proceeds continuously, whether it is a damage or whether it is a lesion. So damage is as it is. So basically there is no repair. Okay. Here I will write no repair of damage. So repair is not done. Thus the synthesis is taking place continuously. Okay. The term which is used, that is bypass. Bypass the lesion or damage. So let's see. First of all, there are polymerase, special polymerases for the translation synthesis. So which are those polymerases? In prokaryotes. In prokaryotes, these are the polymerases. See, polymerase 5, UMUC, UMUD2 plus C. So this is a translation synthesis polymerase in prokaryotes. And in eukaryotes, it is done by this polymerase kappa, polymerase zeta. Okay. Others are as well as polymerase eta. Right. So these are the polymerase in prokaryotes and eukaryotes. So let's see how is the pathway now process. See, this is just an example. DNA damage. So there are two pathways basically. First is the DNA repair. Okay. And the next DNA damage tolerance. In tolerance thing, translation synthesis comes. Okay. So what? Basically this is not a repair pathway. Okay. This is not a repair pathway. It is just continuing the synthesis forward. Alright. Means the damage is remain like that as it is in the DNA. Okay. So damage is not repaired. The replication is continuously done. So this is a damage tolerance pathway. Right. After translation synthesis also repair can take place. Right. We can see here. This can go for a repair afterwards also. Now this is in prokaryotes. In prokaryotes, we know DNA polymerase 3 is important for application, main polymerase. Right. This is a beta clamp. Now here is a damage or we can say lesion. Now what happens? This polymerase, main polymerase released out. Okay, we say that polymerase stall. Okay, replication stops here. DNA polymerase cannot move forward because of this damage. So this is removed out. The main polymerase is removed out. And the polymerase for translation synthesis comes. See, polymerase 4, that is DNB, or polymerase 5. Just now we saw UMUD2C. Okay, these are for the translation synthesis. They will continue the synthesis. See, they will continue the synthesis over the lesion. Right. So the replication is proceeding with the help of this translation polymerases. Right. It will continue till some nucleotides after the damage, okay, or lesion, and the original polymerase will again come, DNA polymerase 3, continuing the further replication. Right. So for a short stretch of damage or lesion, this translation polymerase come. That is polymerase 4, also known as DNB, or polymerase 5, known as UMUD2 or C. So this is in prokaryotes. Very simple. In prokaryotes. Right. Now let's see in eukaryotes. So in eukaryote, there are two models basically. So this first model is polymerase switching. Model. And the part B is the gap filling model. Okay, so these are the two models predicted for the working of translation synthesis. So what happens here? Same like that. See, here is a damage. Okay, this is the main replicative DNA polymerase. Okay, in eukaryotes, we say delta, epsilon are the main. Now what happens? As there is a damage, this clamp known as PCNA in eukaryote is ubiquitinated. See, means what? Degradation will take place as there is a damage here. So they will be removed out. They cannot proceed because of this damage. Then the next translation polymerase will come. Okay, translation polymerase. We saw there are different types. There are 5 types of polymerases. Okay, as we saw in the diagram. Polymerase kappa, polymerase eta, polymerase zeta, right? So anyone can come and proceed the replication. See, this is polymerase switching. Means what? Main replicative polymerase will be removed out. Translation polymerase will come, continue the replication until the damage. Okay. And then again, the main replicative polymerase will come and continue the further. Right. For short stretch of damage, translation comes. Suppose I say this is a, suppose this yellow color is a polymerase. It will just continue till here. A short stretch of DNA damage. And again the main polymerase will continue the further replication. Right, so this is the polymerase switching model. In the second gap filling model, what happens? First of all the synthesis will be done by replicative DNA polymerase. See. Okay. Here ubiquitin is still attached. Here is a damage. This, what it will do? It will stop at the damage. Right. It will stop. And again it will move a little nucleotides further. Okay. It has stopped here. And it is continuing from here again. Right. So this is gap filling model. So first of all, this replicative will continue the replication, and then the next polymerase will come, translation DNA polymerase, and that will fill the gap. So the gap is filled later in this case. So this is known as gap filling model because there is gap at the damage site, and that gap is filled by the translation DNA polymerase after the replicative polymerase has completed the DNA replication, right? And here in the polymerase switching, first the polymerase stops, then translation polymerase comes, and again replicative polymerase comes, but here gap is filled later on. Okay, so these are the two steps in eukaryotes. Now this is a highly mutagenic process. As we saw, this can lead to a lot of mutations because the damage is still present. We can see here damage is still there, right? Damage is still there. Thus this is a mutagenesis, means mutations are still present in the DNA, right? So this is tightly regulated process. Okay, tightly regulated. So this, how is this regulation? Basically, the regulation is known as SOS response. Okay, SOS response. The REC-A protein is very important for this pathway to be initiated. Okay. And these enzymes are not always active, right? Repair enzymes. When there is damage or lesion is present, mutation is present, then only it gets activated. So how is this process? See, first of all, this is in the resting stage. LEX-A gene transcribe the LEX-A protein. Now this is acting as a repressor here. The repressed REC-A gene. So REC-A gene is very important. When there is damage or when there is lesion, the REC-A gene has to be activated. Okay, otherwise it is silent by this LEX-A repressor. Okay, these are the target genes. Now what happens here? This is a induction of REC-A. How is the induction? First of all, activated REC-A. So this REC-A gene is induced. These are the REC-A proteins. Okay, then REC-A proteins trigger LEX-A cleavage. Right, means this REC-A protein will cleave this. Okay, will cleave this. Now this is cleaved, so this cannot be act as a repressor. So these proteins are activated now, and they will responsible for the expression of target gene, or we can say repair pathways. Proteins will be produced, right? Suppose if we say translation, so all the translation polymerase will be produced, right? So this is the SOS response, in which what we saw, two important proteins. LEX-A is a repressor. Okay, repressor means no gene expression. No gene expression is seen when LEX-A is active. Okay, but when there is damage, a REC-A gene is induced and the protein is synthesized, which is a, what we can say, important for repair pathways. And because of this, we can say, we can see expression of got it. So this is all about translation synthesis and SOS response, which is important for regulating this translation synthesis, which is a mutagenic process, as well as it can also control the other repair pathways. Okay, so that's it for this video. I hope this video helps you. If you find this video helpful, do let us know in the comment section.